Okay, thank you for the introduction, and thanks everybody for attending. And this is a joint work with Paul Medvedev, who's over there in the audience. And let's dive right in. So IonTorrent is a relatively new, relatively promising sequencing platform. It's supposed to be pretty fast, pretty cheap, and pretty easy to deal with. And it has been gaining some popularity. We start seeing publications using it. And actually, we were at a talk by IonTorrent, and really liked some of the algorithmic aspects and some of the issues that it presented, so we started thinking about it. So I'll start with a brief overview of the technology, so we'll all be on the same page. You start pretty much as every other sequencing technology. You take your DNA, you break it into small pieces, you add the adapters. At this point, it's rather similar to 454. You mix the fragments with the magnetic beads, these small beads, and you shake it, and you make sure that each fragment is attached to a single bead. So you have beads with templates. Then using PCR, you make sure that you have plenty of copies of the template, DNA template, on each bead. So you have this tube with beads, and on each bead you have many copies of a single DNA template. At this step, you pour your beads onto the chip. The chip contains many wells. So this is one well with one bead and many copies of one template. So this is the template. And is this the pointer? No, on top. Oh, yeah, I got it. Thanks. Okay, so this is the bead, this is the well, this is the template. And okay, so basically, and then the sequencing continues by applying each time a single nucleotide, many copies of single nucleotide onto the chip. And when the nucleotide is the complementary nucleotide of the leftmost nucleotide of the template, they would incorporate. And this doesn't happen here because C is not complementary to T. But in the next flow, you can guess what's going to happen. We have A's, and A is complementary to T. So one A would bind with this T. And this happens for every copy of the template. So we have many such incorporations, each one releasing a single hydrogen ion. This results in a change in pH, which is registered by the pH sensor at the bottom of the well. And we have this peak in pH. Then we continue. We wash the A's away. And the next flow is G's. So the G's are complementary to the C. And here something interesting happens. So we actually have two G's incorporated, because the first one incorporates the leftmost C, and then there's another leftmost C. So another G incorporates. And this would continue for as long as we have C's. So this time we have two hydrogen ions. We have twice the change in pH. And this is also registered by the sensor. So basically, the changes in pH indicate the number of nucleotides incorporated. In 454, it's instead of pH, you have a optical signal. And this goes on. Then we have T's. We register the T's. And then basically, our goal is to deduce the sequence from the sequence of changes in pH. So there are no C's. Then there is one A. Then there's two G's, and so on and so on. So basically, the data looks like this. This is what they call a flowgram of a single well. So we have the flows starting from 1 all through 400. And on the y-axis is the change in pH after normalizing and correcting some noisy effects and stuff. And the letter indicates which base was actually flowed onto the chip. So for example, here we have T, which is roughly at 1. So this is probably an incorporation of a single T nucleotide. And here we have a G, which is roughly at 3. So this is probably an incorporation of 3 G nucleotides. And as you can see, it starts off really promising. And the signal really clusters nicely around the integers. So you can pretty much tell what's going on at the beginning. But as the sequencing continues, for various reasons and artifacts involved in the sequencing, the noise increases and accumulates, and eventually you get this really noisy data. And here it's becoming quite difficult to actually call what's going on. Take, for example, this A here. It's pretty much in the middle. Is this an incorporation, or is it a non-incorporation? If you make a mistake calling it, it's an insertion or deletion, and you don't want that. So basically, the problem of base calling with IonTorrent is taking this flowgram and converting it into a sequence, a read. So IonTorrent do this really straightforwardly. This is similar to what's been going on with 454. They apply a rounding scheme. So for example, we start with this toy example. If the signal is 0.1, we round it to 0 and say there's no incorporation, there's no A. Then if the signal is 2.6, this is rounded to 3. And then we say, okay, there have been 3 copies of this C nucleotide, so now we have 3 Cs in the sequence. And we continue from left to right and construct our sequence. Okay, so this got us thinking that working flow by flow and rounding the signals is not optimal. It's a good approach, but it's not optimal. So take this example. So in this orange row, imagine that the signal is 0.4. So if you go by rounding, you would round this to 0, and you say there's no nucleotide incorporated here. And sort of our key observation was that you could do better than that. So take, for example, in this story, we have an incorporated T 2 flows before, and we have an incorporated T 2 flows after, right? And if you think about it a bit, you will see that this is an impossible scenario, right? Because we said that once we have a bunch of T's, if we have 2 T's, they should all be incorporated in the first T flow. So we should have observed 2 T's here and no T here. So obviously something went wrong in the middle. And we can look at the possibilities in the middle and say, okay, so this one is the most likely to have gone wrong, correct this one, and we saved ourselves from single error. So that's the basic idea. Slightly in a more formal way, we're thinking of a state. So a hidden state is the set of candidate nucleotides. These are the possibilities of the next nucleotide in the sequence. So we start off with all 4 nucleotides, and as the sequencing progresses, we update our state according to the results. So for example, if we don't see an A incorporated at the beginning, so A is no longer a candidate in the next step, and so on and so on. And if you track down this table, you would see that rounding here would lead you to an empty state, which is impossible. You cannot have an empty state of candidates for the next nucleotide in the sequence. So this can be captured, so far it's been an intuition, can be captured in the form of this state machine with a state graph. So the nodes are states, and the edges give transitions between states, and the solid edges are incorporations, and the dashed edges are non-incorporations, so you're losing candidates as you go along the dashed edges. And the point is that any legitimate sequence is translated to a path along this graph. So this is when you apply rounding, and eventually you get stuck at an empty state, because you're at a G and there's no incorporation, so you sort of don't have anywhere to go. And what you really want to do is to find a path that goes all along the state graph and continues downwards. So you don't really want to iterate over all possible rounded values. You want to iterate over all paths in the graph and find the best one. And this would save you from doing such mistakes. Okay, so as statisticians, we need a likelihood framework to solve our problems. So we have this insight of the state machine, the state graph, and we have some insights on the noise of the IonTorrent sequencing process. We want to take all these together and put them in one formula. So we have this likelihood formula. We have the candidate sequence. So for each possible sequence, we have a candidate. We have the sequence. A sequence translates into a series of state transitions. We have the observed incorporation values. This is the flowgram, the noisy values. And then the likelihood decomposes into 3 major components. So we have the transition state probabilities. This is the probability of actually traversing along the graph from top to bottom. We have the priors on homopolymer lengths. This is an idea borrowed from 454. And we have the actual emission probabilities. So this is the noise, actually. So if the true number of incorporations was 3, what is the probability of observing 3.1 or 2.5, the distribution around the true number of incorporations. So great, we have a likelihood function. Let's go over the three components and break them in small pieces to understand them. The transition probabilities are obviously the most important. So assume you have the state with three candidates, A, C, and G, and the next row is C. So naturally, you would say, well, C is going to be the next candidate with probability of 1/3. So I will see an incorporation with probability of 1/3, and no incorporation with probability of 2/3. And you can construct the other transition probabilities in the same manner. And now this is a really flexible framework. So suppose you're sequencing a genome with a very high or very low GC content, you can plug it into the formulas and say, okay, G's and C's are more likely. So if the next flow is C and I have a C as a candidate, it's more likely might be more likely than 1/3. And the emission probabilities are simple double exponential distribution around the true number of incorporations. So if we have 2 incorporations, we expect to see 2, but we have a nice double exponential around 2. And this was actually a comment by one of our reviewers, which turned out to be really, really helpful. We started with a Gaussian distribution, moved to the double exponential. It was actually pretty good. It improved the results. So and the last thing is the prior on the homopolymers. So the basic idea is that, for example, 3 A's are less likely than 2 A's, because 2 A's means 2 A's and then C or G or T. So we have actually 3 possibilities, and 3 A's is just 1 possibility. And in a sense, if you think about it, if you have this rounding method, it places a threshold of 0.5, whether you call it as a 2 incorporations or 3 incorporations. So this threshold sort of assumes an equal probability on all these scenarios. And this is probably not optimal. So once you have a likelihood framework, instead of this rounding ad hoc algorithm, you can plug in any prior you want. So you can plug in a flat prior, which is equivalent to rounding. You can do the geometric prior, which is what has been done by 454. And you can plug in an empirical base prior where you estimate the distribution of homopolymers from the data and then feed that back into your algorithm. So it's a really flexible framework. Okay, then you have to solve it. So now we can calculate the likelihood for every sequence. We want to find the optimal sequence. Obviously, we're not going to iterate over all possible sequences. And because of this nice graph structure, we can solve it with dynamic programming. So we define the Viterbi, use the Viterbi algorithm with some tweaks that make it slightly more efficient than the usual generic Viterbi algorithm. So it's fast, it doesn't take too much memory, and you can run it on as many cores as you like because we're actually processing each well separately. There's no information between wells. So in terms of running time, this is really fast. And then we started playing around with the algorithm. We didn't really like the results. We started investigating, and we figured out that we have something missing. So it turns out that the way the noise behaves really changes from different runs of the IonTorrent PGM machine. And I guess that people who know more about sequencing shouldn't be surprised by that. That noise is a very noisy thing. So anyway, we want a flexible framework to allow for that as well. Okay, so we take the standard deviations of our double exponential distributions, and we model these as a linear function of the flow index. So if you recall the picture of the flowgram from before, we saw that the noise increases as the sequencing continues. And this can be captured with this model. So we allow the noise to increase, and we allow the amount, the increment to change between different experiments. Okay, and this needs to be estimated. We don't have any way to determine this a priori. So we use the forward algorithm, which is basically similar to the Viterbi algorithm. Instead of finding the optimal path, the forward does a summation over all possible paths, and this results in an evaluation of the likelihood function. So great. So you do a grid or a greedy search, and you find the optimal values of these parameters, and then you plug that back into the Viterbi, and you get the maximum likelihood estimates of the sequences, and that's it. And now we have our candidate sequences, which we believe should be better than simple rounding. All right, so we went on to checking it on real data. We downloaded this E. coli dataset from the IonTorrent webpage. It's freely available. Almost 7 million flow grams. It's a standard PGM. Oh, one thing I forgot to mention. Okay, so in all the toy examples I showed you, we had like this very simple repetition over the nucleotides during the sequencing. It was always ACGT, ACGT. And this is how it used to work with 454. Actually, IonTorrent have a much more complicated repetition, a sequence that repeats. It's like a 32 nucleotide long. They have some reasons, I guess, why it's this way. And so we ran our code, Flowgram Fixer, which is, by the way, available, freely available, open source on our webpage. And the IonTorrent calling software. It took under 4 hours. And then we mapped the reads back to the E. coli genome with bowtie. Okay, and these are the results. So basically, at this point, we were thinking about the end user. So this is the biologist who spent a lot of time and money collecting and preparing samples and sequencing them. And he cares about how much information, how many reads he gets at the end. So if he uses the baseline, the IonTorrent software, he would have about 3.5 million reads uniquely aligned to the genome. So this is what he can actually use. Switching to Flowgram Fixer, he would have about 250,000 more, which is 7% roughly, that uniquely aligned to the genome. Another metric for comparison is looking at the map quality of the beginnings of the reads. So this is also a metric that appears in the automatically generated reports of IonTorrent. So this is why we wanted to look at it. So what, so what you actually do is you chop the first 200 nucleotides of the read, and you align them to the genome. And this actually gives the IonTorrent baseline algorithm a big advantage because, as you recall, the noise happens towards the end of the read. So at the beginning, it's really easy to call. So this should be an easier problem. And here as well, we have almost 250,000 more reads, even though both methods succeed with more reads. So it's only a 5% improvement with very high quality reads. And the same goes for different map quality thresholds and different length hard clipping thresholds. Okay, so this was really good. We were very happy with these results. And by the way, I mentioned earlier about the double exponential. So initially we had here like only 5%, and switching to double exponential, which is heavy-tailed distribution, upped us to 7%. So how and why does it work? So what you see here on the left is the error profile. So we have these reads that are aligned to the genome, and we can go flow by flow and calculate the error rate. And what you see is that when you do rounding, you have this very strange pattern of errors. So we have these T's that have very low errors, and these A's and C's and T's over here that have very high error rates. And this is, well, because of this complex structure of the state machine and probabilities, there are some positions which would be harder to call when you do rounding. And what you see on the right is we take this error profile of Flowgram Fixer, and we subtract from that the error profile of the rounding method. So basically, this is the error difference, and so positive is good for us. This means we're winning, and negative is bad for us. It means we're losing. And what you see is that at these places where rounding did very bad, these are exactly the places where we have a considerable improvement. And actually, you have some places where we have a minor, where the rounding beats us by a bit. And we actually have a pretty good idea of why is that, and that's part of a future project. So basically what happens is that on the chip, we use the same noise model for the entire chip, and that's too optimistic. So one thing you could think of is fitting this noise model with the forward algorithm for every read separately. And that's too much, that would take too long a time, but you could say that there's some special structure on the chip because of the way the water washes over it and stuff like that. And we could have a bunch of other ideas. One thing we might do is incorporate quality scores with the forward-backward algorithm. You can get the posterior probabilities at each flow and have like really good quality scores. Okay, so that's pretty much it. One thing I would like to say is that we are looking for collaborations for data. So we're looking for people who are using IonTorrent for short RNA sequencing, microRNAs or whatever, have some neat ideas on what can be done with this kind of data. So if you're interested, please do approach us. And that's it. Thanks to all the funding agencies and to Paul and to you for your attention. Thanks.
Thanks for your talk. Did you try to align with TMAP, with the mapper provided by...
Okay, so that's a good question. So TMAP is the liner by IonTorrent, or yes. It's more suitable for these kinds of reads. So actually we haven't. We had some technical problems with that, and we were under some pressure of time. So we didn't. We felt that like if we align both with bowtie, it's a fair comparison, but definitely it's something that needs to be aligned with TMAP.
Okay.
Yes.
And another question, did it lead to difference between the prediction of the variation? Because, for example, did you have some prediction at the beginning, so without the correction, that you've lost at the end, and did you try to validate the difference between the two predictions?
Between the prediction at the beginning?
So, for example, you had your reads at the beginning, and you had your reads that you improved.
Okay, yeah.
And it may lead to different predictions.
Mm-hmm.
So, did it lead to different predictions first?
So I think this is what we try to capture here.
Yeah, but it's not really prediction. So it's a number of ACGT at all the positions. But did it lead to really difference of prediction of variation?
No, we didn't. But
okay.
That's a good
okay.
point.
And maybe we can talk about that.
Okay, yeah, sure.
Okay, thank you.
So after your collection, the read length changed or not?
They did grow by
you
a
did
bit.
not report. You just say you
right.
had more reads mapped compared to the 454.
It's a good point. We were struggling with there's a bunch of metrics for measuring performance. How many bases you have?
Do you have any idea about how many bases what the percentage you shorten for each read?
So basically what happens is you have the very good part of the read and the very bad part of the read, and in the middle, this is the place where we improve the most. So you would get reads which are longer, but they would be longer by 3 to 7 nucleotides after alignment.
So the reads from your code is shorter, right?
No, a bit longer.
Even longer? And it looks like your hypothesis look like you will get a short reads. You get a longer reads or you get a short reads compared to the original one.
Okay, I understand your point. So what I was talking about is after alignment. So if you take your read
before the alignment, when you call the base. When you call the base for each reads, you have two different ways. Your way is shorter or longer than the original one.
So it's hard to tell because if you place a prior on homopolymer length,
you did not pay attention.
We didn't, but this sort of pulls you towards a shorter read. But on the other hand, the fact that you're using a state machine and prevents you from placing zeros at some places, so this pulls you towards a longer read.
Okay.
So it's hard to quantify, but that's a good point.
I'm just curious if you could expand a little bit how you intended to sort of correct this drop in error right there?
Yeah, so it took us quite a while. So what we think happens is that you're using the single noise model for the whole chip. And then you have some places where you overestimate the noise and some places where you underestimate. And calling these easy places to call here, the easier places are more affected by these mistakes. Because a lot of noise means you're putting more weight on the probabilistic part and less weight on the combinatorial part. And these are the places where the combinatorial part would give you a lot of information. So if you overspecify the noise, if your noise estimate is too high, you're going to underdo these and overdo the difficult problems where the combinatorial part is less important and all of the information is in the probabilistic part. So we believe that adapting the noise to different worlds differently would solve this. Okay, thank you.
Okay, thank you. Thanks, Ben. Okay, so we'll allow 1 minute for people to change.